Hello. Oh, it's working. Go! It's 9:30. No. Somebody yell no. Yes. Oh my God, people. All right. Whoops. No, you'll really be sleeping up there. All right. We'll get the spotlight on the front. All right, guys. Do you have any questions? No? Oh my God. Is anybody breathing?
Barely.
Does everybody have a pulse? Everybody take your pulse. Make sure you have a heartbeat. Okay. Thank you for coming. I'm going to make you play games whether you like it or not. Yeah, no. So our secret word for the rest of the semester, because it just makes it easier for me, is going to be gonad. Okay? So thank you for playing. So I'll forget that it is, and some lectures we may not talk about gonads at all. I try to talk about them as much as possible, as it turns out. But, so. Okay, so today we're going to talk about my favorite thing ever. Yes, it's DNA. I love that molecule. I want to marry it. You love it so much, honey. Give it. Okay. I love it because it's beautiful, right? And it obeys rules and it's logical. I'm all about things being logical, right? And obeying rules. That's my deal. Okay, and DNA always does that, and DNA is the basis for life on this planet, right? We talked about before, every single living thing on this planet uses DNA to store its genetic material, okay? To store the instructions for being whatever it happens to be. If you are a specific type of bacteria, like one that lives in your colon, your DNA determines your ability to be that organism and to survive in the colon, like colon-specific genes. Thank you. I'll be here all semester. You guys are dying. Oh, I'm sorry. Okay, so we're going to talk about DNA today, and that's going to make me happy, and it's probably going to kill you guys, but too bad. Okay. So what is DNA? It is a molecule, right? A chemical. Everything on this planet is essentially an organic chemical, right? Except for rocks and minerals and crap like that. Any part of a living tissue, right? So when we talk about chemicals, we don't mean something synthetic, right? We always think of chemicals like acid that burns you, right? Something in a chemistry lab that gives off smoke and turns blue or whatever, right? So it's the molecule that is passed from parent to offspring that determines everything about you, okay? Everything about every cell, how it's all put together, driving us through development, right? Going from one single cell, right? The egg and the sperm come together. You have 46 chromosomes. DNA makes up those chromosomes. That's what a chromosome is made of. DNA and some proteins that help keep its structure. Okay? The information, the instructions for all the stuff that needs to happen to make an organism is coded for in the DNA. Okay? So DNA is precious. We don't want it to get screwed up. We want to protect our DNA from mutations. Right? Mutations change the sequence, the order of our DNA molecules that can either do nothing, it can totally screw up some developmental process, or it's possible that it can make a new allele, right, and generate a little bit of a new phenotype, for better or for worse. Okay, so we're going to talk about that. Just think of it as the instruction manual for cells, right? And for us, every one of our cells has a little bit different of a job, right? If you're a liver cell, you're doing a different job than if you're a brain cell. Yes, sir? So his question is, if DNA is a logical molecule and it follows the rules, if there's a mutation, what does that mean? Right? Because DNA follows the rules, that mutation can persist through generations. Right? Mutation is the driving force of evolution. Right? If we all started as one single cell in the primordial soup, right, with this genetic material, if there was never a change in that genetic material, if it remained that same exact sequence, this planet would still have just that one organism. Right? The only thing that makes cells different from each other is either what genes they have on and off in, like us, or how different they are from each other. Okay? And we'll talk a little bit about mutation and not all the mechanisms, but when we get to cancer and understanding why that happens, that's all about mutations, and it's all about DNA. So we need to know some of the structure of DNA, how it works, and what its function is to understand when things go wrong, you can almost always extrapolate it back to something happened to the DNA. Okay? So remember, we talked about the cells when I killed you with all the organelles. Our DNA is inside the nucleus, right, in a eukaryotic cell. Right? This is most of the cells on the planet, unless it's a bacteria, are eukaryotic cells. Okay, remember the bacteria are those teeny tiny little guys that just have a cell membrane and ribosomes and DNA, and they don't have these internal little vesicles or sacs. Okay, so DNA lives in the nucleus of a eukaryotic cell. That's where it stays, and it's protected in there. That never leaves the nucleus, the DNA. Okay. The DNA molecule is individual little subunits all linked together. Okay, that's what's sort of being shown here. One little piece of it. All of these are put together. That whole thing is microscopic. It's kind of wrapped up on itself, and then that molecule is wrapped up on itself, and then that's wrapped up on itself until it turns into the little sausage guys that we recognize as a chromosome. Okay, and I'll show you a picture of what those look like under the microscope. We can't see DNA under a microscope when it's all unraveled. Most of the time in our cells, our DNA is unraveled in the nucleus. It's out in its spaghetti form. Okay, that's when it can be used to make proteins and do information that we'll talk about in the next couple weeks. If we want to make more cells, we have to compact down the DNA so we can get the right chromosomes to the right places. Right? Like if you were trying to make eggs or sperm, you have to put all your DNA into your chromosomes so that you can put half one way, half the other. So the egg has 23, and when the sperm comes with it, it has 23, and then you get 46 back. Okay? So this is the DNA molecule wrapped up with some proteins to give it structure in the chromosome like we think of them. And so if we look at chromosomes under a microscope, they look like little sausages. Okay? And they kind of look like they're an X shape. So each side of the X is actually one chromosome and its duplicate. So the only time we can actually see chromosomes is when we've already replicated them or made another copy. Okay? This cell was about to split into two cells. That's the only time you can actually see these under a microscope. And we'll talk about when we do cell division, one chromosome goes to one cell, the other chromosome goes to the other of each of these, so that when you have 46 chromosomes, we make 92, and then we do 46 one way, 46 the other way. You have 2 new cells, each with 46, right? Like if you were in the liver. Let's say you're a liver cell, and the body you happen to live in had a lot of alcohol over the weekend, right? Did some damage. You need to make a few new cells. You would replicate your DNA, all your chromosomes, and then you'd split each one one way, one the other. You'd end up with 2 liver cells, each with 46 chromosomes. Okay. If you didn't first double that number, the 46 chromosomes in the liver, you'd split that. It would be 23. And then if those needed, you drank a lot over the weekend, and those need to make another one, what's half of 23? Oh, 11 and a half at each, and then half of each. Pretty soon, after you've gone through several rounds to repair that tissue, those cells have 4 chromosomes in them. Okay? Every single one of our cells in our bodies needs to have 46 chromosomes in it, except the eggs and the sperm. Those are the only ones that have 23 chromosomes. Right? So anytime I list any type of cell, a brain cell, a skin cell, a liver cell, a pancreas cell, gonadal tissue, outside of the actual egg or sperm, have 46 chromosomes. And we need to keep them that way as we make more and more of them. Right? So this is what your DNA would look like if we took a cell that was going through this cell division that's ready to split into 2. Okay? And you can see that if you count these up, this equals 46, right? Because each set is in a pair. They're still attached, so we still call this one chromosome. Okay? But if you counted these individual strands, these individual guys, then we'd be up to 92. So this is DNA, right? The molecules of DNA all wrapped up and wrapped up and wrapped up and coiled on each other and coiled and coiled and coiled and coiled till they make these little fat sausage guys that we can then see through a microscope. All right. So we call them 23 pairs, which equals 46, right? 23 times 2. And then if you're wondering how you became a boy or a girl, I know it keeps you up late at night. It's all about the sex chromosomes. Okay? This is called a karyotype. It's just whatever your spread of chromosomes is. And this one has an X and a Y. That makes the cell, that the person that this cell came from, a boy. Okay? Girls have 2 X chromosomes. Boys have an X and a Y. The rest of them, pair 22, 21, 20, 19, are all the same between males and females, have exactly the same genes on them. Okay? The only difference lies in this pair right here. And as you can see, this guy's little. He's little bitty, little baby, little guy. Okay? So the genes down here, some of them are the same on the X and the Y. There's a few that are the same down here. The Y is missing all of these up here. So if you're a boy, right, you only have 1 copy of every gene. You only have 1 allele of the genes that are on the X chromosome. Right? All of us have 2 alleles on the other ones, right? If you're a gene on chromosome 19, right, that gene and that gene are exactly the same. These are the same chromosomes. It's a pair. One you got from mom, one you got from dad. So like earlobe-shaped gene, if it's on chromosome 19, say it sits right there, You got one from your dad, you got one from your mom. You have two alleles of that gene. Maybe attached, or attached, detached, right? But it's the same gene you have two of. So every single gene in our body we have two copies of, if you're a girl. And if you're a boy, you have two copies of every gene except the ones that are on the X chromosome. You only have one. Okay? And as a little bonus, there are some extra genes on the Y that do not exist on the X, and that's what makes you a boy. The maleness genes. Okay? It's called SRY. That's what drives testosterone, and that's what drives the development of the male organs. By default, female is the default sex. Sorry. We're just the default. That's sad. So if you have an X and a Y, but the Y is not functioning, you have a mutation in the SRY gene and it doesn't work, you appear to be a female. Okay? You won't be a fertile female because to be a fertile female you need two X's, but phenotypically by outward views you look just like a girl. Okay? So two X's is female and X and a Y is male. Go! It's time already. I don't know what...
Boy! Thank you, sir.
What the hell?
Blue, you're my boy!
Uh-huh.
Thank you, sir.
Oh, yeah. Forgot about that. Okay, let's do a little top hat quickly, just because we can. Hopefully it's relevant, because you never know. Yo! Look at that. Ah, what the heck? I'm having some issues. There we go. Are we all in view? Okay, all the correct answers, more than one could be right. Right? So you should be allowed to pick more than one. You should be allowed to, if you're texting, you put two letters, if there's two or three. So A, B, C, D, if all of them are right or whatever. I don't know, it's an experiment. Let's see if that works. It says it's supposed to work. What? How do you put two answers? I don't know what you're asking. Is it working answering two? Okay, there's two right answers. I'm just going to tell you that. Yes? Yes, when we get to genetic section, we're going to talk about things that go wrong. Yeah. Yeah, like XXY and X, yeah. Yeah, we'll get to that too. So, anything that's not an egg or a sperm, okay? Every other cell type in our body, how many chromosomes do we have in there? Humans, right? Because all organisms have their own little number. Remember a good friend, the donkey, had 62? Yes? It won't let you send two? Are all the texters you can only answer one? Or did you do it? You did it okay? Is it good? Oh, you have to, no space between the letters? You guys are following directions, that's okay. Oh my God, everybody's sleeping today. Wake up. Okay, are we ready? Not yet. Oh my Lord. We're all going to get this right, correct? Yes? No. Yes. No, I think they want you to put they only want this. So if you, like you would put A and B right next to each other. I think that's what they said worked. If it happens to be. How's it going over here? You guys are good? All right, let's view our answers already. Ooh, there's somebody peeking in there. Hello. Are we ready? Oh, I can't even see. What the heck's going on? I don't even know how to look at this now. Oh, okay, that's the top. Okay, that's right. 46 chromosomes or 23 pairs. Right? 23 chromosomes is only correct if we're talking about sperm or eggs. Okay? How come this keeps going off the screen? What the heck, people? Look, it's just going off there. It doesn't want to stay. Right? 46 pairs is way too many. Look at us getting more and more knowledgeable. If you have too many chromosomes, most of the time you do not make it through embryonic development. Okay? Plants are one of the very few organisms that can have multiple copies of their entire genome. Like instead of having 46, they could have another 23 on top of that and still be okay. But animals can't tolerate that. So one too many or one too few is almost always lethal. There's a few examples of where you can have one extra chromosome and survive and be kind of okay. That would be an example. That would be Down syndrome, right? Down syndrome individuals have 3 chromosome 21s instead of just 2, right? So they have severe developmental delays. They have lots of problems with their organs, heart malfunction, lots of stuff. Lots of them do not survive to birth. Okay, so having too many or too few is generally a disaster. And so that's why when we talk about how we get which 23 go one way and which 23 go the other, when we're talking about making eggs and sperm, it's incredibly important that the correct ones go the right way and that it's 23 and 23, not 22 and 24. All right, we've had enough of this for now. Maybe later. Why is this so crazy today? It's all crazy. Oh, that's not the right one either. Okay. All right, so let's talk about DNA in a little bit more specifics because this is where the logic comes in. This is how we can understand why when mutations happen, it can be bad or good or neutral, right? If we understand what DNA is made out of and how it's built. And so DNA is composed of individual molecules called nucleotides. Each one of these is depicted as that guy right here, this guy, right? And they're in a long chain, and there's 2 separate chains that are attached together. Okay? So they call it a double-stranded molecule. There's 2 strands of these nucleotides attached together, and they're always attached to each other. Okay? So DNA always exists as a chain of molecules on one side, a chain of molecules on the other side, and they're bonded together by chemistry in the middle. So the stupid little dotted lines is a chemical bond just called a hydrogen bond. Okay? If 2 nucleotides of the appropriate type come near each other, they have to stick together. They don't have a choice. Okay? The rules of chemistry and physics on this planet determine that those 2 stick together. So they can't not if they get too close together. So that holds these 2 strands together. Okay? The information is held in the sequence of these different little chemical structures called nucleotides. Okay? There's only 4 nucleotides that make up DNA. It's the order that these are found in that determines the difference between a gene for insulin and a gene for building some other kind of hormone, right? Or a gene for making a cytoskeletal protein to help build your cells. It's all about that order of the nucleotides. Okay? And each nucleotide, which I don't really care if you know, actually is made up of some sugar. So this is actually like a little sugar molecule. They call this guy a base. He's just the guy that sticks in the inside that does the chemical bonding. And then the sugars are attached through this phosphate group. Okay? But I'm not going to ask you that because you're not biology majors, as it turns out. Okay. So we call this, it looks kind of like a rope ladder, DNA does. So each one of the red guys, right, that's an individual strand, and then they're depicting, right, those bases that stick in with that bond as the rungs of the ladder. And it ends up being twisted, and again, that's just based on chemistry. Okay, so this side is one strand, this side is another strand, right, and they're connected together in the middle. Okay, so 2 single strands of these nucleotides in specific order, and they're attached together. Because it's double-stranded and they're stuck together like that, that makes this molecule very stable. Okay, so that's one of the importances of DNA. That's what makes it a really good storage material for information, right? It doesn't break down very easily. If it was just one of those strands, then it would be easy to clip it apart, and then that would not be something that you want to store all the information for making you as a human being or any organism, because it could easily get destroyed and then you're dead. Okay, so it's a relatively stable molecule. It can still be damaged, but it's much more stable than some of the other molecules out there. All right. Like I said, there's only 4 different types of nucleotides in DNA. Okay. We are going to refer to them as A, T, G, and C. Their real names are adenine, thymine, guanine, and cytosine. It's just the kind of molecule they are, whatever. It's just how they're named by chemistry. Okay? That's all we have. So it's just the order of those that can create all the different genes out there. And when I said DNA is very logical and it makes a lot of sense, there's only 2 different ways we can attach those 2 strands together. Okay? If there's an adenine or an A on one of the strands, the other strand has to have a T to stick together. The A will not stick to a C or a G, right, by the chemistry crap. It's just the wrong shape. You can jam them as close together as you want to. They will not form that stable bond together. The A's and T's, if you stick them near each other, they make this stable structure. Okay? If there's a C, it can only bond with the G. So if there's a string of C's on one strand, there's a string of G's on the other strand. Okay? So if you look at one single strand of DNA, you know exactly what's on the other strand just by this stupid rule. So this is a rule you have to know. A's and T's stick together and G's and C's stick together. So if I ask you, one strand of DNA has AAACC, the other strand has TTTGG. You'd be able to tell me that because of this rule. You will be able to tell me that, right? Yes? Thank you for playing. Does that make sense? That's the only rule. Okay? You have these 4 A's and T's go together, G's and C's go together to build that double-stranded molecule. Okay. As we go on talking about cell division and cancer, you'll see why this is one of the most important rules on our planet as far as keeping organisms alive and well. Okay? It's called complementary base pairing. It happens to be one of my favorite things in the world. Yeah. No, that's a great question. And an A cannot bond with an A. They cannot bond with themselves either. Okay? So A's and T's always together, G's and C's always together. Occasionally, if weird shit happens, they can, but it's always bad. It's always would be considered a mutation if that occurred somewhere in the DNA. That would mess everything up. And we'll talk about when that kind of crazy stuff happens as we go forward and talk more about cancer. Okay? Cancer is all about those bases being changed and messing with the sequence. Okay? A specific gene has a specific sequence. If the gene is supposed to start AAT, CCG, and you change one of those, that can change that gene completely. That can make something wrong. Either that could kill the cell or cause cancer or something else bad could happen. Okay? A 1 base pair change can cause disaster. A 1 base pair change in the gene that makes hemoglobin, who knows what hemoglobin is? It's now your favorite protein. Hemoglobin carries oxygen in our blood. How important is carrying oxygen in your blood? Yeah, because what happens if you don't carry oxygen? Yeah, you're dead. Right? That's pretty important. Okay? Hemoglobin it's this really complicated protein. It uses iron to help you carry oxygen. Right? So if you're anemic, you don't get enough iron, eat more spinach. Right? You'll be tired. Oh, I'm not getting oxygen to my brain. 1 base pair change in that molecule messes up the hemoglobin protein enough that it causes sickle cell anemia. If you've heard of sickle cell anemia, it's when you don't carry oxygen very well. The protein makes this weird shape. It causes your red blood cells to look like a sharp sickle. They get stuck in your bloodstream, and you could die. Okay? Because you can't carry oxygen. If you don't get oxygen to your brain, it only takes 2 to 3 minutes to cause brain death. Okay? So that's just one of these. A G changed to a C. You're fucked. Okay? You guys woke up back there? What? Did she just say what? Ooh, that was on lecture capture. Okay. So this is sort of a close-up of that. I'm not going to ask you specific questions like this, but I want you to understand just why it's so important, right? So here's our 1 strand. They're only showing 2 nucleotides. Okay, they're showing a G and a C on this side, right? This side is the other strand. It's a G and a C, right? So G's and C's. Here's that stupid chemical bond between them, right? So a G with a G is not quite the right shape to fit in there, okay? And a C with a C is not quite the right shape. The T's and the A's certainly aren't the right shape to make this stable bonding, okay, to hold those 2 strands together. Okay. And so this was all determined by our good friends James Watson and Francis Crick. Watson and Crick were the, not discoverers of DNA, but the ones who determined its structure, right? And that allowed us, that structure of how G's and C's go together and A's and T's go together, helped us understand then cell division. How do we make more DNA that's exactly correct to make 2 new cells? Right? Because like I said, if you mess up 1 base pair, it could be disaster. So how do you control not messing up 1 single base pair if you're going to copy 46 chromosomes, which have billions of these stupid A, G, C's, and T's? Right? So it's extremely important to know what the structure is to make sure when we make new cells, they do it correctly. And they almost always do. We make a few mutations here and there, but not very many because of, right, G's and C's and A's and T's, and that's the only thing that can stick together. And so these guys, as it turns out, were really smart people that really didn't want to do a lot of work. Okay? So they spent a lot of time talking to everybody else that did do a lot of work. Okay? And so they used information that other scientists generated. They drank a lot of beer in pubs in England. That's what they did back then, right? They just sort of, and they'd talk about everybody else's data, and they came up with this idea of how it worked. The people that actually did the experiments came up with the wrong structure, right? So the moral of that story is drink more beer when you're thinking, right? It helps you be more creative. Open your mind. I don't know. But they really did drink a lot of beer. Okay, so these are two of the most famous scientists, won the Nobel Prize, published in the best journal on the planet, which still is. Okay. So again, why do we care about DNA structure? I know you guys don't, but you know I do. So I'm going to make you guys understand it too. Okay. That complementary base pairing, the whole idea of G is the complement to C, right? It's G's soulmate, right? If you're complementary to something, that's the one that, you know, makes you whole. I don't know. Okay? So complementary base pairing allows us to make new cells with exactly the same DNA as the original. Because all we have to do is look at that DNA molecule, right, and look at the bottom strand. If it's AGCT, then all we have to do is make the opposite, TCGA. Right? We can just copy by doing complementary base pairing. Now you have the exact second strand, and then you use the top and make the bottom, and you have an exact copy without any mistakes. Okay? That's how DNA replication works, which is what we're going to talk about in the next lecture. Okay? Right? If we don't do this correctly, we're dead. Right? If you start as one single cell with 46 chromosomes, you need to replicate those 46, make 92 exactly like those 46. Put 46 one way, 46 the other. Now, you're 2 cells in the fallopian tube, right? Each one of those splits, now you're 4 cells. Then you're 8, then you're 16, right? And then you make that stupid little blastocyst guy I showed you last time, and you implant in the wall of the uterus, and the miracle of life. Okay? If you don't complementary base pair correctly, a bunch of mutations happen, you don't get past 2 cells. Okay? Blah, blah, blah. We'll talk about this when we get to what's the point of all this information and how do we actually do anything with it. Okay? And because I love it, that's why you guys have to. Okay. All right, so we're going to come back to that. Because I'm not sure what might happen at 10:30. We'll see. Okay, so let's top hat. I'll give you some hints.
Hey, Mr. Grumpy Gills. When life gets you down, you know what you gotta do?
I don't want to know what you got to do.
Just keep swimming, just keep swimming, just keep swimming, swimming, swimming. What do? We swim, swim.
Dory, no singing.
Oh, 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 oh,
Dory.
I love to swim. And
Dory.
when you want to swim, you want
See,
to
I'm going to
keep
get stuck now with that song. Now it's in my head.
Sorry.
Oh, I love Dory. Okay, my friends. Oh, gosh, why is this so crazy? Got some craziness happening here. People! No! Can't even read this shit. Okay. What are the base pairing rules? Look in your notes, people! Everybody's going to get this right today. Or you're going to see me jump up and down and have another hissy fit. Or I won't have a hissy fit. How about that? If everybody gets it right, I'll have a little attack in the front. For your viewing pleasure. Right? What is complementary base pairing? What's happening up here, people? I haven't been up here all semester. Stupid lecture capture. I have to be up front. I requested a spotlight and a moving camera, and they said no. So can't the camera just follow me around? Then I get a little spotlight. How's it going up here? You guys are good? All right. Oh, you guys are so good. Ooh, it's fun up here. All right. Yes, yes. I like what you've done here. Mm-hmm, yes. I have a t-shirt like that. My honey badger don't care. He doesn't give a shit. He eats larvae. So nasty. Oh! Thank you. You're killing me. Okay. See, I was up there lounging with my friends in the back. All right, how do we feel, guys? 99%. Where's that other percenter? They're not here. They're sleeping in. How's everybody's texting going? It's awesome? Okay. I am still selling Girl Scout cookies. In my $4. Holy crap. And guess what else I'll be selling soon? World's finest chocolate. My son's school's doing World's finest chocolate for the athletics program. So I'll have my case of chocolate. I'll probably just eat it all and owe them $60. Be like in my office, that looks really good. It's good. The chocolate with the almonds. Yeah. And the chocolate caramel, I fall for that one too. I can't. I love chocolate. No, I think World's Finest only does it through fundraising stuff. I remember when I was a kid, they were like enormous, huge, fat. Now, they're the same price, but they keep getting smaller. And they used to have like a McDonald's coupon on them or something. I don't know what these will have.
Pizza Hut.
Is it Pizza Hut? Oh, I will let you people know. Okay, I'm looking for 100%. What do you think? Oh, crap, we can't see it. No! Who's doing that? You did that on purpose. It must be a texter because they didn't change their answer. Ah! They want to see me go like this. A's and T's, G's and C's. Live it, learn it, love it. Okay. Okay. Man, why is my... Okay. Okay, we're going to come back to this because, again, I don't know what's going to happen. Maybe something might happen at 10:30 and maybe not. It's unknown at this point. Everybody. Okay, so we'll wait and see if anybody shows up. All right, so we're going to start the next lecture because, and because a couple of lectures when we start talking about cancer are a little bit longer, so we want to have time to make sure if you guys have any questions for that kind of stuff, because I know you guys are so full of questions all the time. I'm going to start requiring you to ask questions back there. Talking to you back there in the back row. They're like, whatever. Is that woman, she's still talking? What's going on? Okay. So replication, this is what I was alluding to, right? Making an identical copy of the DNA so that you can put identical copies into new cells, right? That's the whole point. That's why complementary base pairing exists, to make sure that happens correctly. Because again, a one base pair change could be a disaster. Now it depends on where it is, right? A one base pair change in a particular spot on the hemoglobin gene causes disaster. But a one base pair change or mutation in some other genes might not do anything, right? It's sort of luck of the draw, okay? But you don't want to risk making a whole bunch of mutations if lots of them are going to be super detrimental, especially during embryonic development. You want everything to go correctly, okay? When things go wrong, embryos die. So we want to make sure we make the exact right DNA, the exact chromosome number 1 copied exactly. Okay, and there's billions of nucleotides in all these chromosomes. So we have to be careful, and we have to have a method to make sure it works. And the method to make sure it works is this whole stupid complementary base pairing. Okay? And so we have a double-stranded molecule. Right? That's how it's and it's complementary base paired. All the A's on the bottom strand are bonded to T's on the top strand. All the G's are to C's. All the C's are to G's. All the G's are okay? In order to copy this guy, we have to pull those two apart just transiently, just for a little bit, in order to add new nucleotides to make two new double strands. I'm going to show you guys a little educational video about how this works, and it's going to say a lot more details, and it's going to mention all this shit that you guys don't need to know about. But the little video helps you understand the process and why this stupid complementary base pairing is so important. Okay? And so we pull these two stupid strands apart, and then we have an enzyme called DNA polymerase. I do want you to know the name of that enzyme. Poly means lots, a mer is that little unit, and ASE means it's an enzyme. So anytime you see a word with ASE on the end, it's an enzyme. Okay, polymerase, ligase, I don't know what else, lactase. Okay. So DNA polymerase makes more DNA. Okay. It polymerizes the nucleotides of DNA together to make another DNA molecule, to make another chromosome. Okay. Again, complementary base pairing. If this polymerase is following along the strand and there's a G sitting here, it will jam a C, right? Do that stupid bonding for a C. If there's a T, it sticks an A. If there's a C, it puts a G. If there's an A, it puts a T. It's an idiot-proof way to make a new strand. Okay? Yes, mistakes happen, but very rarely. So occasionally, an A might get jammed up against an A. We usually fix that because it doesn't fit exactly right in the strand, and it makes a weird shape. And our little enzymes go, oh, crap, I need to proofread, and this doesn't look right. They can pull it out and put the correct thing there. But occasionally, the wrong thing gets through, but very rarely. Okay? Usually, something gets through. It adds T to an A, but the T was damaged by something, like UV irradiation or some sort of radioactive material or some other craziness. Okay? Some chemical carcinogen. Okay? Smoking, something like that. Okay? So the sequence G, 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 C, 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 A, 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 T, 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 T on the top, C, 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 C. It's just stupid. Oh, if it's a G, I have to add a C. Oh, if it's an A, I add a T. Right? And just follows it along. On each side, on the top makes a new strand. Now there's a new double strand up here. That's a chromosome. A new strand on the bottom. Now that's a new double-stranded. That's the other half of that chromosome in that X shape. So his question is, if DNA is damaged, is there any way to fix it? Yes, we do have a bunch of enzymes that help fix DNA. And so if mutations get through in this process, we can fix most of them. Okay? If we didn't have all those, we would have cancers all over the place. We cannot fix, his question is, what about Down syndrome? We cannot repair, like, the addition of an entire chromosome. Right? So that's more than, when we talk about a mutation like a single nucleotide mutation, that would be a chromosomal mutation. We have one whole extra chromosome 21, right? Unless that, if you did in vitro fertilization, you have that one egg and you could pull that extra out at that point, that'd be the only way that you could fix that, right? So we just want everything to work correctly, right? Sometimes chromosomes are damaged, they're missing a whole chunk. We can't fix that. Right? We can use complementary base pairing, what's on one strand to fix another if DNA, if the actual nucleotides are damaged by UV irradiation. So UV light, the reason we talked about skin color and race, UV damages especially Ts. Okay, it messes up particularly this nucleotide and knocks chemical structures off of it. If they don't get fixed and we do replication, polymerase doesn't recognize it as a T because it's messed up, and we'll add some other, right? It'll go, I don't know what that is. I'll put a C there. And then you persist with these mutations. If you have a problem with the repair of UV damage, you have xeroderma pigmentosa, which are the people that can't go out in sunlight at all. Because if they go in sunlight for 10 minutes, they will get skin cancers all over them because they can't repair damage. Okay, so yeah, we do repair lots of damage. Sometimes some damage gets through, right? Because people do get skin cancer, but if you didn't have damage repair, we'd all have skin cancer just walking from your car to this room. Yeah?
So that would be a mutation that could get passed on to their children?
So the question is, could that mutation get passed on to their children? The only mutations that can get passed on to your children are the ones that occur in the eggs or the sperm, right? So if you're missing the gene or the gene is damaged that does the repair, that gene's damaged in all your, if you were born with that, then you're going to pass that, right? But if you just get damage in your skin, right? Let's say, you know, I've got too much UV and I'm not using sunscreen and I get a skin cancer right here, that damage to that DNA is not passed to my offspring, right? Because that's only in this skin cell, not in the gonads. Now, if you lay out in the sun and put your, no, right? So that's why when you're at the dentist and they want to do x-rays, they put the big thing of lead over you. It's all about protecting your ovaries or your testicles. Now that we've said testicles, let's talk about, now that somebody's brought up testicles, okay, yeah. So her question is, what if skin cancer runs in your family? Like everybody in your family, not everybody, lots of people in your family have had skin cancer. Does that mean you're at higher risk? It's a great question. It depends on whether it's based on the environment that your family's been in and how they've, you know, they laid out in the sun with the big silver thing with no, you know, they put baby oil on and they've just been out there their whole life, right? Or it's possible that your repair mechanisms work, just not as well as they're supposed to, right? So there is something, there's a one base pair change in your repair mechanisms that make it, it's pretty good. It's not 100% efficient. So it's more likely at some point something won't get fixed. Whereas the person next to you has 100% efficient repair mechanisms, they're going to fix it every single time, right? But even if you have that little bit of damage, doesn't work quite as well, but you wear sunscreen and a hat and you don't go to the beach and lay out all the time, you're very careful, and the person next to you is just baking, every day in baby oil, they're at higher risk than you. So it's the environment and sort of what you're born with, right? So things that run in your family generally are something in your DNA that's either a mutation or it's just not working as well as it should, right? Because there's a little bit of a change. Not enough to give you the disease where you can't be in the sun at all, but you're at higher risk because your repair doesn't work as well as somebody else's, right? Does that make sense? Okay, we'll talk more about hereditary cancer too and how mutations can play a role in that as we get closer to that stuff as well. Okay. So this is just the little picture of what I was talking about. So we separate, so there's our piece of, little tiny piece of the chromosome that we've opened up. Now we break apart those base pairs, the A, G, C's, and T's on one side and the other side, and we do complementary base pairing all the way down that strand. DNA polymerase, if the red strand, if that's a T, it added an A. If it's a G, it added a C, and so forth, and just follows it along, right? Just by those stupid complementary base pairing rules, right, it makes an exact copy, right? If this is a T, that's an A. If that's a G, that's a C. Which means if this was a G, this one over here had to have been a C, right? Which means now this one has to be a G. So this new strand is identical to this old strand because these two were originally complementary. And this new strand is identical to this old strand, right, because these two were complementary. So we've made this exact molecule is exactly like the original, and this one is exactly like the original, right? And so this chromosome, when you finish the whole thing, would go one way, and this chromosome would go the other way, and then you'd separate the cells, and now you have cells with the exact same DNA in them, right? This would happen on all 46 chromosomes. We'd copy all 46 chromosomes exactly. This 46 would be over here in a cell. This 46 would be over there. Now you have two new liver cells with the exact same DNA, right? No mistakes. Exact same 46 chromosomes here, exact same 46 chromosomes over here. Just because of that stupid complementary base pairing. The idiot-proof power of that process. Okay, so we're going to watch DNA replication. Again, they're going to talk about a whole bunch of details, but they're going to show you how these stupid little nucleotides come in, and hopefully that will help make a little more sense. And again, I'm not going to ask you super details. I do want you to know what complementary base pairing is, who goes with what. If I give you one strand, you can tell me what the other strand looks like. And that DNA polymerase is the one that's doing it.
The rule of base pairing helps explain how DNA is replicated prior to cell division. Enzymes unzip the DNA by breaking the hydrogen bonds between the base pairs. The unpaired bases are now free to bind with other nucleotides with the appropriate complementary bases. The enzyme primase begins the process by synthesizing short primers of RNA nucleotides complementary to the unpaired DNA. DNA polymerase now attaches DNA nucleotides to one end of the growing complementary strand of nucleotides. Replication proceeds continuously along one strand, called the leading strand, which is shown here on the left. The process occurs in separate short segments called Okazaki fragments, next to the other, or lagging strand on the right. This difference is due to the fact that DNA polymerase can only add new nucleotides to the 3' end of a nucleotide strand. A primer begins any new strand, including each Okazaki fragment. An enzyme replaces the RNA primer with DNA nucleotides. Then an enzyme called DNA ligase binds the fragments to one another. There are now 2 DNA molecules. Each consists of an original nucleotide strand next to a new complementary strand. The 2 molecules are identical to each other.
It's still going. Riveting! Okay, and so then if we remember back to our picture of the chromosome, that karyotype, right, that would be one half of the X, that would be the other half of the X. Okay? So that was a whole lot more detail than you need to know, right, about the primer, and one's going leading and one's going lagging. But the basics of sticking in the stupid nucleotides based on complementary base pairing, right, off of one strand, off of the other, to make 2 identical new chromosomes, essentially, right? And then one can go one way and one can go the other way. We only do DNA replication if the cell needs to divide. If the cell doesn't need to divide, right, it's just a skin cell and it's just happy-go-lucky in the epidermis, just enjoying life, it's not going to do DNA replication. It just wants those 46 chromosomes to do its job of being skin. Okay? If you burn yourself or cut yourself and you need to heal over that, you have to make new cells for that, right? That's what makes the healing. So we have to do cell division. We'd replicate and then do mitosis. Did you talk to anybody else? No. I think they're all sleeping. Are you game? Okay. All right, come on up. I'm proud. Would you like a... Okay, here's a bag. I have all colors.
Well, I guess I don't really need a different hair color, do I?
I'm going to... Oh, I don't have my phone. I need a rub. Let them see what we're up to. Okay, I told you this was going to be a show. I said I wasn't sure if there was something going to happen at 10:30 or not, because I didn't send out the email until this morning. And Cheryl's probably still in bed.
Well, what about the rest of the dumbasses?
The rest of the dumbasses are dumbasses. So, all right, I'm going to do this. Okay. You sure you don't want to go orange?
Oh, sure.
Okay.
We'll go orange.
I'm going to go into the changing room.
Perhaps purple is too subtle.
Okay. There, how do I look? Is that good? What am I doing? Hot? Okay, so if you want to be off of lecture capture, you have to be beyond the table. If you want to be in lecture capture, then you're right here.
Oh. I am happy to have my foolishness
Okay.
be recorded for.
All right, this is audience participation, please. So feel free to get up and join in.
Yeah, right.
I know, they're like,
They're just all going to stare at us.
do you think this is educational or not? It's so educational.
6 years of graduate school, and look at our boy now. Oh, Randolph. Now, I remember somebody that wasn't too different from this not too long ago.
Okay, here we go. All right. Ready?
Enzyme, what you doing today? I said enzyme, want to make DNA. I said enzyme, show me your fighting way. Let me mix you with some primers. Enzyme, time to build a new strand. I said enzyme,
Sing it, people!
I know that you can. I said enzyme, these letters also spell Dan. All we need to get things started. It's just a
To all the scientists out there doing PCR, Bio-Rad salutes you with the all-new line of SoFast Supermixes for real-time PCR.
Right. So that's a real commercial for nerdy science stuff.
I don't need to go work out this afternoon.
This is Dr. McElprang. She's a microbiologist here at Seaside. She's joining us. I want to thank her for joining us today.
You are very welcome.
All right. We're singing in your class next week?
Yeah, next week.
Okay.
Sometime.
Sometime next week? All right. Thank you. Aw, that was sweet. She's my baby sister. Oh my God, now we got to go back to this. What time is it? Is it time to go yet? Almost! Yay! Yes?
Can you actually, you know how in your video it showed us the process of every phase? Is this something that you can actually see in the microscope, or is this a conclusion that was brought up?
You can, so our question is, in the video where you see all this happening, can we actually see that under a microscope? No. So all of the stuff that we know, now, and remember what we said about science, it's all about a hypothesis that can be supported. We can't really prove anything, we don't really know anything. We extrapolate from mostly biochemical experiments, right? So based on all of the experiments that have been done on replication and how it works, it's our best guess at how it works. Right now, we're getting really close to being able to see DNA at the atomic level. It's called an atomic force microscope. It's really expensive. But we're getting closer to being able to actually watch something like this happen. But right now, we haven't been able to yet. So it's a guess based on all the experiments that have been done. Yeah? No. Aww. Okay. All right. All right, my friends. Okay. So this is the bottom line of replication. Again, only when cells are about to divide. Right? Otherwise, we don't want more chromosomes in there. More chromosomes are bad. We only want the exact number we want. Okay? Human beings, we want 46 in every cell. If you're a donkey, you want 62, or horses is 64, whatever. That's your number, right? We don't want 47. If we have 47 and that extra one is chromosome 21, you have Down syndrome. Okay? We want exactly what we want. Right? So each new chromosome is half of the old one of the strands, and then a new one that we built. The other one's an old one, the template, and a new one that we built. Okay? That together is that X-shaped chromosome that we saw in the karyotype. And then we pull those apart so one goes each way. And then the new cell has 46 and 46 and the correct 46. Right? Having 46 is great, but if it's, if you have 4 chromosome number 1s, you're screwed. Okay? We want to have the correct number of everything. So here's our little picture, right? Here's our little sausage chromosome. We can almost never see them like this because they don't squish down till after replication. But if they did, right, it would be all, right, it's a cajillion billion of those nucleotides, and then they're wrapped and wrapped and wrapped, and then that's wrapped on itself, and that's wrapped on itself, and that's wrapped on itself, until it would look like this little sausage. Okay? So when they're all out like this, that thing would split. We'd build a new half on that side, a new half on that side. Right? It would all wrap down on itself, and then it looks like that little X-shaped guy. So that's the cartoon of the photo karyotype that we showed before. Okay? A duplicated chromosome, a replicated chromosome. The thing that attaches in the middle they call the centromere, so you can think of it like in the center right? Each one of these is called a sister. So this is a sister to this one. You can think of it as an identical twin sister, okay? Because they're the exact same sequence, right? This double strand on this side is the exact sequences as this strand. Has the exact same alleles, the exact same genes. Everything's exactly the same. Why did they name it sisters instead of brothers? Well, I don't know. Men probably named it, so I don't know. They like the sisters better. It's interesting because most of the stuff that we talk about is all in the female or in the feminine. Like they call what you have a parent cell, and then it splits, you know, the DNA, they call those daughter cells. So, yeah, I don't know. They all go with the whole female thing. I don't know. Okay. Go! Again, we are on fire. What's going on here? Let's see.
Dong. Dong.
Dong, Grandpa is talking to you.
Dong. Where is my automobile? Automobile?
Leg. Big leg. Why, you little scuzzbag. Whoa!
Oh, Long Duck Dong, you are so funny. Who knows what movie that's from? Woohoo! Good job, people. If you haven't seen it, go watch it. Late night TV. It's got to be on a rerun on some channel. Okay, what did I just say we were doing? What are we doing? Oh, this one. Oh, my God. This one, this one. Oh, maybe this one's hard. Let's see. Let's give you, don't answer till I give you some hints. And you can actually see it. Okay. All right, so replication overall is copying one of the strands using complementary base pairing, copying both the strands of DNA of that double-stranded guy by complementary base pairing, both the strands just randomly, haphazardly, like idiots, or one strand just haphazardly. Both strands or one strand, complementary base pairing, or just shit luck. Oh, and I did forget to mention, right, after the first 2 minutes, I'm not going to say if, when you guys would like to see the exam questions and what you got wrong, I have all of your Scantrons in my office, and I have copies of all the different versions. You can always come to office hours to look at them to see what you got right and what you got wrong. The final exam is cumulative, so this stuff will come back to haunt us again. So I want to make sure you know which concepts you're more familiar with or less familiar with, so those things you can concentrate on studying later. You can come and see those at any time during the semester. Okay? If you can't come to office hours because you have class, shoot me an email and give me some windows of time that you're around so that I can be available for you. Okay? I'm always here. I'm just not always